I did my undergraduate studies at University of Toronto, where I was a chemistry and biochemistry major, and went on from there to do a Ph.D. at Albert Einstein in New York, where I focused on molecular biology and biochemistry, and then did my postdoctoral research at UCSF in San Francisco, where I started studying cell biology and developmental biology. The primary focus of my lab is split amongst 3 different projects, where 2 of them we study the development of the fruit fly embryo, where in one project we study how cells change their shape during development in order to make different tissues and structures. The other area of interest in fruit flies is understanding what happens to cells when they die and what regulates their death in order to accommodate changes and defects that occur during development. The third area of work in my laboratory is more in technique development and innovation, where we develop new techniques in developmental biology and in proteomics. One of the more valuable contributions my lab has made in the world of proteomics is the discovery and development of a technique called difference gel electrophoresis, or as we call it, DGE, or the rest of the world calls it DIGE. And in this method, we compare 2 or 3 protein samples by putting different color fluorescent tags on the proteins, and then mixing them all together and running them on the same gel, and analyzing the 3 different colors of the fluorescent dyes that we're looking at. This was an idea that came to me when I was a first-year graduate student at Albert Einstein College of Medicine. And in those days, the technology didn't exist for making these dyes and for imaging the dyes and for even doing the mass spectrometry analysis of the proteins that we discover. It wasn't until I moved to Carnegie Mellon that, working with Alan Wagner and a chemistry graduate student, that we were able to synthesize the proper sets of dyes and develop the imaging tools and the analysis tools to make the system work. My lab is composed of 4 graduate students, 1 technician, and a person who used to be a postdoc in my lab who's now faculty over at Pitt, and we're co-directing a project in the laboratory. In addition to that, there are 3 or 4 undergrads always working. The individual projects that are going on in the laboratory are, one student is developing a new method for studying how cells change their shape during development. And this particular project is very exciting because we're using a new type of reagent called killer red, which is a fluorescent protein that, when irradiated with light, destroys the function of the protein to which it's attached. And that way we can ask about individual proteins at specific points in time and space during development to see what their role is in any particular process. Another project in the laboratory is trying to find out what happens to cells when they die and other processes within the cell that lead to the degradation of the cell, and in particular autophagy. And we're creating new reporters that allow us to follow cells over time and monitor when they die or when autophagy occurs and what happens to the breakdown products of those 2 events. A third major project in the laboratory is studying the role of a small heat shock protein in cell death. We used a proteomics method that we developed in the lab to look for protein changes that happen early in cell death events, and found that this heat shock protein was changed. And that when we knock this protein out, it gets rid of all cell death in the embryo. So we're trying to understand how that protein works. A fourth project in the lab is more on the inventive side of the lab, where we're building a new imaging device that allows us to detect proteins over a 1,000,000-fold concentration range. This device outstrips any existing devices by more than 50-fold, so that we'll be able to peer more deeply into the proteome and discover protein changes that happen at a very small scale that will be important for disease detection and for understanding development in cell biology. The techniques and tools that we use in my lab are quite varied, ranging from organic synthesis and development of new fluorescent dyes, all the way up to developing new microscopy tools and techniques and computational analysis of time-lapse image movies. The main techniques that we use in my lab are time-lapse microscopy of embryo development, fluorescent reagents, and also the development of proteomics tools. The journals that my lab publishes in are Science, Development, Journal of Cell Biology. The types of questions that we're hope to address in the next 5 years or so is continuing on the research that we're doing and understanding how cells change their shape during development and what regulates that. And an important message in that is that there are dozens, if not hundreds, of different proteins that are required for maintaining and establishing a cell's shape. But for a cell to change its shape during development, only a small set of those proteins are actually worked upon or physically changed, and the rest just sort of follow along. And using the proteomics tools that we've developed, we want to delve more deeply into understanding what the real targets of the developmental cues are with respect to how cells change their shape. With respect to cell death and engulfment and autophagy, we want to continue to develop new tools and understand how cells during development are, when they die, how they're removed and what signals their death, and understanding other processes that change the internal structure of the cell, such as autophagy, are regulated during development. In terms of technique development and the entrepreneurial side of the lab, we're working very hard to expand the capabilities of looking as deep into the proteome as possible, so that ultimately one day we can look 25 million, 50 million fold deep into the proteome so that we can see protein changes that are indicative of specific diseases. So people that have graduated from my lab have a wide variety of careers right now. Some are still in academia, teaching at small colleges or universities. Others have gone on to get their degrees in law and have become patent lawyers. One is now a lawyer at the FDA. I think the reason why students would be attracted to our program is several reasons. One is interdisciplinarity, in that our laboratories, even though there's a small number of, relatively small number of labs in the department, we cover a huge variety of modern molecular biology and cell biology. Another important aspect of our program is that the lab sizes are a manageable size, averaging from about 6 to 9 people per laboratory. So there's good access to the head of the laboratory and ability to learn from more senior graduate students in the laboratory. There's also access to undergrads, where every lab has several undergrads, so that gives training opportunities for the graduate students to learn how to direct other people in research. My favorite part about Carnegie Mellon is simply the open door policy that we have here, in that we all try to do interdisciplinary research, and in order to have that work, we need to have students from different disciplines. We need to have collaborations from different parts of the university. And I have had graduate students in my laboratory that have come from all across the university, from the business school, from statistics, from mathematics, from computational biology, from robotics and engineering. And so it's been, and chemistry. So it's been a wonderful experience to share ideas and develop ideas, taking advantage of the great expertise across the entire university without any barriers, without any impediments in establishing those collaborations.